So with that, let me talk about the PDB very briefly. This is the first open access digital data resource in all of biology. It was established in 1971 with just 7 protein structures. It's grown to become the single global archive for protein, DNA, RNA, experimentally determined structures of biomolecules. The current holdings of the archive now exceed 230,000 structures. The archive is managed jointly with our worldwide protein data bank partners in Europe and in Asia, plus the team at the electron microscopy data bank and the biological magnetic resonance data bank, both specialist repositories for 3D EM and NMR data. At the foot of some of the slides that I'll show, you can see citations of 2 relevant papers. So the PDB is crucial for sustaining the broader biodata infrastructure. It's a single, fair, fact-compliant global archive that provides open access to public domain experimental 3D biostructures. All of our data are distributed under the most permissive Creative Commons CC0 license, with which we actually distribute data coming from 3 data centers in North America, Europe, and Asia, loading on any given week somewhere between 300 and 400 new structures to the PDB archive. We've been recognized by the Core Trust Seal and the Global Biodata Coalition and funded by NSF, NIH, and DOE. We have 5 interlocking services at RCSB Protein Data Bank, the US Data Center for the Worldwide Protein Data Bank Partnership, and these 5 services, numbered 0 through 4, turn global data into global knowledge. We have impact across the biosciences, literally from agriculture to zoology, serving the awardees of NSF grants, NIH grants, and grants from the US Department of Energy. Our RCSB.org website is a one-stop shop for public domain 3D biostructure data. We offer access to more than 230 experimentally determined PDB structures, alongside more than 1 million computed structure models coming from AlphaFold DB and the Model Archive. This combination ensures that we can provide 3D structure information for all of the proteins constituting the proteomes of human, the model organisms, select pathogens, and crop plants, important crop plants, and organisms that are important in understanding the impact of climate change. We have a series of exploration and visualization tools, many of which you'll see today, courtesy of Sebastian. They work equally well with the experimentally determined PDB structures and the computed structure models coming from AlphaFold DB and the Model Archive. And just to be clear, the provenance and reliability of both types of data are clearly identified. The Erlenmeyer flask denotes experimental structures. The video display monitor denotes computed structure models. This is probably the most important slide that I'll show. This is the homepage of the RCSB.org website. And what you'll see inside this red ellipse is the toggle that allows you to opt in to actually get access to all of the computed structure models that we deliver alongside the 230,000 plus experimentally determined PDB structures. So let's move on to the star of today's show, which is influenza A virus. The first recorded avian influenza outbreak, described as foul plague, was documented in 1878 in Italy. I want to draw your attention to the fact that the WHO has announced nearly 1,000 deaths, human deaths due to influenza A H5N1 virus, 1,000 cases, killing approximately half of those individuals. That's a very disturbing case fatality rate for any infectious disease specialist. On the 6th of January of this year, the first US avian influenza death was described. This involved an individual who had a backyard flock of chickens that presumably contracted the influenza A H5N1 virus from a migratory bird, such as a duck. The anatomy of the virus is depicted here schematically in a compelling image produced by David Goodsell. It's an envelope virus with negative sense single-stranded RNA segments, numbering a total of 8. There are two major surface glycoproteins, hemagglutinin with a number attached to it, neuraminidase with a number attached to it. There's an ion channel, an RNA-dependent RNA polymerase, which is a heterotrimer. There's also a nucleoprotein present within the capsid and an M1 matrix protein. And you can see shown off on the right here some of David Goodsell's renditions of the atomic level 3D structures of components of the virus. The virus life cycle is depicted here. The hemagglutinin is essential for getting in. The RNA-dependent RNA polymerase both supports viral mRNA production and genomic RNA synthesis. And the neuraminidase is responsible for facilitating release of the virus from an infected cell, allowing it to go on and infect other cells or other individuals. There are enormous advantages for influenza viruses having segmented genomes. Different segments encode different genes. And when you have co-infection of a cell with two viruses, you can actually create a new virus with admixtures of the different components, shown here initially in red and black. In the host nucleus, you've got a mix of both red and black RNA segments, and then a new virus with an assortment of these RNA segments can be produced. Hence, all the different variants that you're aware of, H5N1, etc. And I also want to draw your attention, of course, to the fact that point mutations occurring in different messenger RNAs as a result of errors made by the RNA-dependent RNA polymerase can also cause changes to the viral RNAs and hence the viral proteins. So just a word about nomenclature and specifically why H5N1. The standard way of referring to an influenza virus is to first enumerate the type, then its geographic origin, the strain number, the year it's isolated, and then the virus subtype, which is defined by which hemagglutinin is present in the virus and which neuraminidase. And each one of these hemagglutinins and neuraminidase have distinct numbering. So H5N1 is very much in the news. We are seeing human cases in the US and other countries. These are principally impacting workers in poultry farms and on dairy farms, but the big concern is that these viruses may mutate further to allow H5N1 to pass readily from human to human. Very recently, a team at the Scripps Research Institute led by Ian Wilson showed that as few as 2 very well-placed mutations will allow the current highly pathogenic avian influenza viruses to become readily transmissible between humans. The situation could be much worse than we saw with SARS-CoV-2. And notwithstanding some of the news that we're hearing from the NIH about their potential unwillingness to continue mRNA vaccine research, I think it's clearly the case that mRNA vaccines could be critical in combating a further mutated H5N1 virus that can be transmitted between humans with a potential case fatality rate as high as 50%. So I'll turn over now to Sebastian Bittrich, who is a longtime member of the team at UCSD and the San Diego Supercomputer Center, and he'll show you how we can use tools at rcsb.org to explore H5N1. And I think I've already stopped sharing. Looks to me like I've already stopped sharing. Sebastian, over to you, sir.
Thank you for the kind introduction, Stephen. So for the second part, we want to provide a more hands-on experience and have a look at the biological story that Stephen just presented and use visualization-related tools on RCSB.org to have a closer look at some of the key proteins and key processes that are part of the virus life cycle. And here you can see a simplified view of the viral life cycle with the core proteins and the 3 steps of the virus particle getting entry to avian or mammalian cell, replicating its RNA as well as proteins, and then releasing new virus particles that can infect other cells in the same organism or spread to other organisms. And in particular, I want to have a look at 2 proteins, hemagglutinin, and have a closer look at what determines host specificity. So what's necessary for hemagglutinin to bind to sugar moieties exposed on the surface of a host cell. So what establishes this binding? And then we can try to understand what determines specificity. So what would be necessary for H5N1 to switch specificity from avian host cells to mammalian or even human cells, and get some insights into what would be necessary for potentially human-to-human transmission of this virus, which would be a pretty dire situation. And at the same time, or as a second part, for some positive news, I think it would also be nice to have a look at existing treatment options that are in our arsenal when it comes to treating influenza A. And I want to have a closer look at zanamivir, which is an inhibitor for neuraminidase activity. And as Stephen stated, this is essential for the release of new virus particles. And if this can be inhibited, that's a reasonable treatment option. And as stated, zanamivir binds to the active site canyon of neuraminidase enzymes. And using tools on RCSB.org, we can search for those binding sites that are necessary for the binding of zanamivir in neuraminidase enzymes, and can find similar arrangements in other proteins coming from the whole PDB archive, as well as the 1 million computed structure models that are integrated to basically answer 2 questions: whether there are other virus infections that could be potentially treated with zanamivir just because they have similar binding sites that should be responsive to a treatment with zanamivir, as well as if there are human proteins where zanamivir might cause side effects. So whether there should be or could be off-site or off-target binding sites that are present in human proteins. And we'll use the structure motif search tool to have a closer look at that question. And with that said, how the second part is structured, we prepared a number of slides that we will share with you after this event if you're interested. But I'll use my browser to navigate to a number of features on rcsb.org, and you can follow along, but you can also try to work these examples side by side if this is more convenient for you. But we'll have a look at some of the core processes, and we'll use that as a chance to have a look at some of the functionality that Mostar offers on rcsb.org. And these slides also contain a number of screenshots just to help you with navigation, as well as links that you can then revisit later on at your own pace. So again, I think it's more convenient to just follow along my screen share, but up to you how you want to work these examples. Okay, first things first, what I'd like to have a closer look at is just how you can access the Mostar 3D viewer and also cover some basics. So most of the time you will start exploring rcsb.org by running a query using this top search bar, where you can use a free text search, for example, for influenza A, to navigate to certain structures of interest. And you can also provide entry identifiers that might be known from a publication to navigate to what we call a structure summary page, which is a nice overview over all the information that is associated to one particular PDB entry. And of course, there's a lot of information provided here. But I want to highlight this panel in the top left corner, which usually provides information that is relevant for the visualization of this protein as well. So in particular, I want to highlight this section where you get this 3D rendered cube as well as the tagline Explore in 3D, which always goes to the Moster 3D viewer and allows you to visualize this entry in 3D. So let's have a look at some of these options to visualize this structure in a generic fashion, a way of highlighting or visualizing sequence annotations. And maybe let's pick electron density. You can also map validation report data onto a 3D structure. You can also see that this hemagglutinin structure has some symmetry present in its entries, so we can visualize that as well. And let's scroll down a structure summary page and have a scan for more of those 3D rendered cubes. So we have a dedicated visualization for glycosylation sites, which might be present in some structures and are certainly relevant for hemagglutinin. And there's also a dedicated section on oligosaccharides where you can get a view on particular instances of those glycosylation sites and focus on a particular chain instance. So let's open one of these views as well. And this is the first view that I'll now navigate to, called structure. So just a generic representation of a structure. And this is how most looks like. So a really convenient tool to visualize 3D structure data directly in your browser without the need for any installation. And like other viewers, you get a 3D canvas where you can rotate your protein, you can zoom in and zoom out and just explore the structure. And by highlighting or hovering over individual residues, you get this tooltip in the bottom right corner, which makes it easier to navigate and see which exact position you're currently looking at. You can also use this sequence panel up top, where you can hover over individual residues, and there's a bidirectional mapping between the sequence view and the 3D structure. So it's a pretty convenient way if you're, for example, looking for a particular sequence position that is known from literature to be crucial for protein function, to navigate and explore that in 3D. And last but not least, I would like to highlight the functionality of Moster, where you can click on any one residue, which allows you to zoom in on that residue. And now you don't just see this generic cartoon representation of the overall polymer chain, but you also see this ball and stick representation of the surroundings of the amino acid that we just clicked on. And you also get those dashed lines that showcase or summarize non-covalent interactions that, for example, can stabilize secondary structure elements or be highly relevant for the binding of ligands, which we'll also explore in just a moment. And last but not least, there's a lot of functionality offered by Moster, and you can find most of it in this panel on the right-hand side with different sections on, for example, visualizing density, which we'll have a closer look at, as well as assembly symmetry. And if you want to learn more about those basics, some of which I don't have time to cover, we have dedicated help resources, which I can highly recommend if you want to learn more about Moster and its functionality. And there's also a video tutorial as well as a dedicated webinar where one of our colleagues explains Moster in great detail and provides a really nice overview over the core functionality of the Moster 3D viewer. So next up, the second link that I initially opened from the structure summary page is called Sequence Annotations in 3D. And again, you get this 3D of your protein of interest that you can have a look at. And you get this bidirectional mapping between sequence and the 3D structure, but this time You don't get just a sequence panel provided by Moster, but you get a lot of information that is attached or mapped to the sequence. And for example, you can now explore secondary structure elements as well as annotations from external resources. And by clicking on these structure elements, you can efficiently navigate to different parts of the structure that might be of interest to you. And there's a lot of information in here. So for example, also regarding validation of this structure where certain chains interact with other chains or ligands, and where you can, for example, see disulfide bridges. So a lot of information to explore here. And there's also an upcoming office hour for this functionality if you want to learn more. This is how the electron density view looks like, which is available for most X-ray diffraction entries as well as electron microscopy entries, and basically looks like the previous 3D representation of a protein. But now you get this density panel expanded, and if you click on any particular region, you get the same view as previously, where you see the surroundings of this selection that you just clicked on, as well as the non-covalent interactions, but you also get everything enriched with density data. And for X-ray, you can also toggle the visuals for those individual channels, as well as adjust the contour level. So again, I think it's a pretty convenient way to access this data directly in your browser with minimal setup. Moving on, this is how the symmetry of this entry is visualized. So here you get access to a dedicated color theme that makes it more obvious which elements are repeating and which elements are unique. So unique elements are colored in distinct colors, but you can see, for example, there is a threefold symmetry here, and certain entities actually have 3 chains that are present. You can see the symmetry axis running through the structure. And all of that, again, is available from a structure summary page by following links, as well as via elements in this panel on the right-hand side, if you know where to look. And lastly, I would like to highlight the bottom section that was called oligosaccharides on the structure summary page, that allows you to zoom in on particular glycosylation sites. So here you can see sugar moieties that are attached covalently bound to the protein chain, and it also uses a dedicated visual language that describes carbohydrates in this abstract via abstract shapes, which makes it a bit easier to parse the concrete sugar monomers that are present in this chain. So this was one example where we had a closer look at hemagglutinin, and the second protein that I want to have a closer look at is neuraminidase. So let's just navigate to its structure summary page. Again, overall, the structure is the same of how information is presented, but this time we have not just oligosaccharides present in this structure, but there's zanamivir, an interesting ligand or inhibitor. And now you get a ligand interaction link that allows you to directly go to a view that showcases how this ligand is positioned in the active site canyon of neuraminidase, and which interactions are formed to the surrounding amino acids. And you can see the hydrogen bonding pattern. And Moster also provides information on less common interaction types, such as pi-cation interactions, which you can see up here, where it's not between the protein and the ligand, but within the protein, there are, for example, interactions between this arginine residues and this tyrosine up here. And again, all this information is readily available with a single click, basically. And for the sake of completeness, if you scroll down after the protein chains are described on the structure summary page, you now get information on small molecules just because there are ligands present in this neuraminidase structure. And you can get additional information on zanamivir as a drug if you follow this link. And like before, you can focus on individual environments, the surroundings of zanamivir if you're interested in that. And we also have some external annotations. So for example, from BindingDB that showcases binding affinity and inhibitor values if they are relevant and available for certain protein structures. Okay. With that said, I want to have a closer look at hemagglutinin, and in particular, the one story, the study that Stephen alluded to by the Wilson lab, where they wanted to investigate what would be necessary to switch host specificity, the recognition of avian receptors, over to ability of hemagglutinin to preferably bind to human sugar moieties or have a preference for the infection of human cells. And a nice way of doing that is not to look at individual structures, but we also have dedicated tools at hand where you can compare structures, for example, by a structure alignment. And from this study, I know two PDB identifiers. One is a structure as it was seen in cattle that were infected with bird flu found in Texas. And then the Wilson lab created a mutant that has oligosaccharides as they would be present on human host cells present, as well as they try to engineer mutations that switches over this recognition from oligosaccharides that are specific to avian species over to human oligosaccharides. And so the first one is basically what was found in this Texan cattle, and the second one is the structure that contains this mutation and is able to infect human cells more efficiently, or has a preference for them. So this is how an alignment looks like. You, as in many other pages, you get this 1D and 3D integration. You can zoom in on the sequence alignment. Overall, you can see that these structures are effectively the same in terms of sequence identity as well as overall structure as quantified by the root mean square deviation as well as TM score, which is not really surprising because there's only a single mutation present here. And now you can use this to compare the alignment, and you can also use this sequence panel to navigate to the position of interest, which is position 226, where the mutation is actually present. And we can have a closer look at this environment. So on the right-hand side, you can see the viral hemagglutinin structure, and then this ball and stick representation represents the oligosaccharides that are exposed on the surface of a host cell. So the one in this ochre color is the avian type, and then in gray you can see the human analog to that. So one interesting observation is that you can switch on this high-level representation of carbohydrate symbols, and just by visual inspection, you can see that the individual monomers are the same, or you have the same elements present. However, their relative arrangement differs, and that can be explained by this linkage, which is different between the avian oligosaccharides versus the human oligosaccharides. But you can also see that up here, where the monomers are exactly the same, the overall structure is the same, and the interface formed between the viral hemagglutinin and those sugar moieties is the same. You can see a pretty clear resemblance of interaction networks, as well as the overall structure, which again not really surprising if you just introduce a single mutation. However, after this linkage differs between the human receptor analog in gray and the one in ochre, you can see that different interactions are formed for the second part. So in particular, I want to point out this hydrogen bond, which is formed between a hydroxyl group of the sugar moiety and a glycine present in the receptor capable of binding to human cells, versus the one that was found in the cattle population, which also has a glycine, but you can see that no interaction can be observed. So probably just because the distances are different, and also you can see that, no, sorry, you can also see that this mutation, of course, has an effect on how this binding site is structured. So in the one that is specific or capable of infecting cattle, you can see a glutamine residue, which forms a quite extensive interaction network to polar functional groups of this sugar moiety, which is just not present if you mutate it to a lysine residue, which is smaller, so it just doesn't reach that close to those functional groups. And it's also just providing methyl groups, so there's no polar functional groups available to even establish hydrogen bonds. So maybe that's also a reason why this relative orientation can be observed, and why there is binding between this glycine backbone and this hydroxyl group that is present in, or that would switch specificity over to human receptors. So yeah, with that said, there's a couple of more material prepared that you can have a look at your own pace. But the second big part that I want to have a closer look at is how we can treat infections and how neuraminidase as an enzyme binds to zanamivir, and answer this question whether this binding site occurs in other viral proteins as well as human proteins. And again, a nice starting point for that is a structure summary page. So if you go to the entry of neuraminidase structure, the one that we had a look at before, and you zoom in on this zanamivir binding site or the active site canyon, you can have a closer look at this interaction network and try to pinpoint certain interactions that you deem highly relevant. It's also helpful to compare it with other, like the natural substrate of this enzyme, as well as other inhibitors that might be known, where we also have a lot of structured data available in the PDB archive. And one thing that stands out is, especially after this comparison to other inhibitors, is the binding or the surroundings of this carboxyl group, where you can see 3 arginine residues that form an interaction network with this carboxyl group. And we can now try to search for arrangements of 3 arginines that have this relative arrangement in terms of distances, as well as relative orientation, how they are positioned with respect to one another, under the assumption that similar arrangements of arginine residues have the same binding capabilities to interact with zanamivir, as well as similar ligands. And we could hypothesize that those 3 arginines are basically a signature for neuraminidase activity, and can run queries that will retrieve those arginine residues. So what you want to do for that is use this panel that is called structure motif search, which allows you to run these queries, and you switch on the selection mode by clicking or toggling this button, which now provides you this menu up top where you can select individual atoms or whole chains, but the default is residues, and that's exactly what we need to search for residue patterns that are reoccurring through the PDB archive. And now if you click on a residue, it doesn't move around and you don't see the interaction network, but you get this halo and can select individual residues in the Moster viewer. And it also populates this list on the right-hand side. So just by clicking on those 3 arginine residues, you can see them occur in this list, and then you can click Submit Search, which will run this query and screen the whole PDB archive for similar arrangements of these 3 arginine residues without directly considering sequence similarity or overall structural similarity for this protein. So we just care about those 3 arginines. And once you run this query, you can see how this type of search is structured, that we point to a particular entry of interest and then have a number of residues that we search for. So in this case, everything is in chain A, and we see those 3 arginines that we just clicked on in 3D, but you can also have interaction motifs between different chains. And you can also observe that these individual arginine positions are quite far apart at sequence level. So again, we're just looking for 3 arginines that are present in the archive, and we get around 800 hits, which is a lot of information to go through. So just for your understanding, I want to highlight the first result item, which is just the identity, the structure that we set out or used to define this query with. So you can see that this matches with a root mean squared deviation of 0. So again, this just means it's the identity because everything is exactly the same. But you can see this list of matched residues. And you can also see that because there's this 4-fold symmetry, that you get 4 instances of this motif that was found in this particular entry. And as you scroll down through this result set, you can see that the RMSD value increases more and more. So the dissimilarity between the identified residues and the ones that we use to define this query increase more and more. And again, there's a lot of data to go through here, but just to wrap things up, I think a really nice way of having a closer look at result sets is the refinements panel, which gives you this breakdown of experimental methods or the taxonomy, which basically answers this question that we set out to answer, whether there are other viruses that contain arrangements that are highly similar to the one from the neuraminidase, and might be also receptive to a treatment with zanamivir as a drug. And you can see that we see influenza A viruses being identified, also subtypes of that, other disease carriers like bacteria, and also other influenza types like influenza B, unidentified influenza somewhere in here. And you can basically scroll down and have a closer look at this result set if you're interested. And another nice feature is that this also allows you to refine your queries and to narrow down into certain subqueries that you might be more interested in. So the second part of the question was whether there are human proteins that have this binding site, and it's fairly easy to have a closer look at that by selecting Homo sapiens and then running this query again by clicking this button up here, which basically enriches this previous query for structure motif with this extra condition of just being interested in human proteins. And now you can see that we get a smaller result set, still a lot of data to sift through. And you can also observe that these hits aren't necessarily human proteins in the sense that a human protein would be identified that contains those 3 arginine residues, but oftentimes you can see interactions between viral neuraminidase that contains those free arginine residues that happens to interact with a human protein, and they are part of the same entry. So like our search system doesn't make a direct connection between this criterion for being a human protein and containing those free arginine residues. And a nice way to make this judgment call is just looking at this static rendering, where you can basically see those 4 neuraminases that we previously identified from the viral neuraminidase. But just by visual inspection and scrolling through this result set, we will be able to find some structures that are different. And in particular, this one stands out, which is actually a human sialidase, so a human neuraminidase, which has a highly similar binding site, which isn't exactly surprising because the biological function is pretty much the same. And interestingly, there's also an inhibitor present in this active site canyon of this neuraminidase. And by clicking this align in 3D link, you can basically bring everything full circle and go back to the 3D view and have a closer look at how this alignment looks like. And we can draw some final conclusions from this. So how this view works is you can see the original motif in green. So this is the viral neuraminidase structure that we extracted this motif of free arginine residues from. And you can see the alignment to this human ciliadase, where you have also those free arginine residues. They are highly similar in their relative arrangement, which is also emphasized by the RMSD value being just 0.5 angstrom, roughly. So those are quite similar. And you can also see that the surroundings or how those arginine residues are positioned, there are stark similarities between these structures, just because they have the same biological function. You can also see both ligands present in this active site, so you can see zanamivir as well as a related sugar analog, which also inhibits the human protein. You can see some differences between the concrete inhibitor that's present. And lastly, you can zoom out and just compare the overall structure. So you can see that the viral protein has this 4-fold symmetry, whereas the human protein, there's only a single copy present in the biological assembly. And you can also make the observation that around this active site, the structural similarities are really pronounced. Whereas as you move further and further away, you can see additional loops being present in either the human protein or the viral protein. So there seems to be more evolutionary pressure acting on the active site and the biological function, of course, versus parts that are further away and not directly related to the function of this protein. And you might also get some indication on why there's only a single copy of this protein chain being present in the human entry, because the human entry in blue has certain loops that interfere with the interface that can be observed for the viral neuraminidase. And last but not least, if you want to have a look at just the global alignment of these structures, you can use the pairwise structure alignment app again. Just provide these identifiers and compare them. And you can basically make the same observations that there's certainly a lot of similarity around this active site, just because the ligand binding capabilities seem to be highly similar and the overall function is known to be similar. But you can also see that at sequence level, there are quite some differences, that you have regions that are only present in one or the other structure. Sequence identity is fairly low, but at structure level, you still see a clear signal that these are the same fold in terms of RMSD, which is fair value at 4 angstrom and a TM score above 0.5 indicates that this is a shared fault. And yeah, that's a brief tour of some of the functionality that's built atop the Moster 3D viewer on rcsb.org. And just to conclude what we had a look at, so we explored individual structures using Moster, then we used the pairwise structure alignment app to relate individual structures to one another and highlight the importance of a single mutation that switches hemagglutinin receptor preference from avian host cells over to human. And we also identified related structures using the structure motif search tool, which revealed similar centimere binding sites. And to wrap things up, so this is just a small part of a larger comprehensive course tour of the proteome of influenza A H5N1 as part of a publication that has been accepted in Protein Science and should be published soon. And big shout out to Shuchi Dutta, one of our colleagues, as part who initiated this manuscript and was pivotal in writing this really nice case study of where we have a look at basically all of the proteins that are relevant in this context and have a look at some more Moster features that I didn't have time to go into more detail in the context of this event. And at the same time, I would like to thank the whole RCSB PDB team, because of course this is a cooperative effort. In particular, I would like to highlight Christine and Shamara, who make sure that this event and many other events run smoothly. We are grateful for federal funders who allow us to advance science as well as provide data free of charge to other academic groups as well as companies, so they can advance science as well. We are grateful for our worldwide protein data bank partners that contribute to data standardization, the relevant formats, as well as some of the software. So for example, PDBE contributes greatly to the development of the Moster 3D viewer. And last but not least, I would like to thank you for your interest in this event, as well as tools that are available on rcsb.org. And with that, we should have time for some questions, if there are any.
Thank you very much, Sebastian. So there are a few questions in the chat, in the Q&A rather. So if you'd like to go back to your rcsb.org page, Luigi Di Costanzo, a former member of our team, wanted you to re-show how to generate the side-by-side visualization of the molecular structure from Molstar and the track representation from the structure summary page.
Are we talking about the sequence annotations in
Yes,
3D features? So there are multiple ways how you can navigate to that functionality. Again, I think it's really convenient to use this panel up here. You can.
Why don't you click on that panel and just show Luigi?
So if you follow this sequence annotations link,
yeah,
this is the view that you get, where you have the 3D view and then all the annotations that are present at sequence level. And again, you can click on individual features, either in the 1D representation or in 3D to have this bidirectional navigation between both representations of a protein structure.
Thank you, Sebastian. There are two technical questions from anonymous attendees in the Q&A, which we'll address in writing. And let's move on to Barry Grant. So Barry Grant asked, will the 3ARG motif search include computed structure models or just PDB entries? And the answer is with the toggle on for okay, Christine is going to type an answer here. And Luigi says, thanks for your answer to his question. He also asks, can a particular motif search be uploaded from an external PDB file? Can the binding structure motif search be extended to AlphaFold models or uploaded PDB files?
And just go back to, sorry, just to go back to Barry's question, Sebastian, can you show the motif search and how to include AlphaFold models with that?
Yeah, sure. So again, for the neuraminidase, just zooming in on this binding site and selecting the 3 arginines that we previously identified. So you would just start with that, run your query, and then you get this toggle that Stephen presented, and it's also present below the advanced query builder. So you need to enable this, run your query again, and you don't get 800 hits that are just from the PDB archive that we originally saw, but you will see also 150 hits that are from computed structure models that are now included. And usually they appear not directly in the beginning, but as shown before, you can also use the refinements panel to refine your query and just focus on these 150 result items and have a closer look at those.
The logic underpinning presenting the experimentally determined structures first is that they are, on average, more accurate than computed structure models, which are typically only as good as lower resolution experimentally determined structures. And they also frequently contain, the structures also frequently include bound ligands, none of which are present in the computed structure models from either AlphaFold DB or the model archive. And so what you can see now that Sebastian has toggled to the computed structure model hits only from the structure motif search is related enzymes, the sialidases, and if you scroll down a bit more, we might see some other enzymatic functionality. Obviously, there are a lot of sialidases encoded by the human genome and the genomes of various model organisms. So these are going to show up in the results from the computed structure models. There are reports in the clinical literature that cross-reactivity of neuraminidase inhibitors with off targets encoded by the human genome may be responsible for some of the side effects that are seen in cases of individuals who've been treated with neuraminidase inhibitors.
So you can see some other functionality, but also this is at really high RMSD values. For just those 3 arginines being aligned, a value of 1.3 is usually not relevant. So
Great. And so Barry Grant is now so Barry has a related question that pertains to computed structure models. So what you'll see in, could you just go to the structure summary page for the AFQ57? Sorry, could you scroll back down again,
sorry.
please? Stop there. Okay, stop there and click on 62576 F1.
This one?
Yeah, that one. Thank you.
Sure.
Okay, so what you can see are 3 blue domains here depicted by Molstar, and then some extended polypeptide chain and some other somewhat organized elements of secondary structure, but they're color-coded orange or yellow. And the reason they're color-coded orange or yellow is because one has to be cautious about these. These are the parts of the polypeptide chain that could not be reliably modeled with, in this case, it's AlphaFold, the AlphaFold 2 calculation. These may well be regions that are with low PLDDT scores that are actually intrinsically disordered in the absence of other protein partners. So when you're looking at a motif like this, at a structure like this, the blue parts of the structure with high potentially reliable PLDDT scores are the parts of the structure that one should be focusing on to try to draw inferences from the AlphaFold 2 computed structure model. The atomic coordinates of the yellow and orange segments of the computed structure model are not to be trusted and should not be relied on. They may well, as I said, be parts of the protein that are intrinsically disordered, which would suggest that you would need to use experimental methods other than either X-ray crystallography or electron microscopy to study those parts of the protein to generate additional hypotheses that one might then go on to test with functional assays, perhaps. So Barry asked if we could exclude the portions of the protein with low PLDDT scores of the computed structure model with low PLDDT scores from the structure motif search calculation.
It's actually the only way. Everything in yellow and orange is ignored just because it's not really necessary. It's not really relevant information. So the relative placement of individual residues is not meaningful. So if it's not structured, you don't want to look for motives. So
okay,
it only works in cyan and dark blue.
Yeah, you're absolutely right. So I want to emphasize that these computed structure models are of very high value where you have no other experimentally determined 3D biostructure information available. Where you do have experimental structures in the PDB, you want to be using those in preference to the computed structure models. And this is why we are presenting on rcsb.org both the experimental structures and the computed structure models. For plant molecular biologists, for example, we want to give them full coverage of the proteomes of Arabidopsis thaliana, soybeans, maize, rice. We know that structural coverage, PDB structural coverage of the Arabidopsis thaliana proteome is only 4%. So in order to provide full coverage to plant molecular biologists, we are providing access to the computed structure models for the other 96% of the Arabidopsis thaliana proteome. I see that we're actually beyond time, so we should probably draw this to a close. I want to thank Sebastian for a terrific job today, Christine, Shamara, and for all their support. And I also want to acknowledge the important role that Shuchi Dutta played in putting together that protein science paper. She's a senior author that served as the template for generating this webinar. And finally, I thank all of you who've joined from probably all over the US and some countries outside the United States. And we're very grateful to host you today, and look forward to seeing you use rcsb.org even more effectively than you have been able to before. Thank you.